Thank you very much, Dr. Peng, for the introduction, and thank you for selecting our paper for the highlight talk. So this is a published work led by my PhD student, Heather. So in this work, we want to study the hidden variable inference problem in molecular QTL mapping. So as a background, what is the QTL analysis? So this is a traditional term traced many years back, but in the current setting, we refer to molecular QTL, which means that the trait is a molecular trait, such as a gene expression. And we want to study the association between a genetic variant, for example, a SNP, and a molecular trait like a gene's expression. So this is commonly referred to as the eQTL analysis, with E standing for expression. And the purpose of doing this is to explain why many GWAS variants are in non-coding regions and try to infer what molecular biology roles those variants may play. So eQTL is the most widely studied analysis, and thanks to the data generation by the GTAC consortium. And besides eQTL, we have SQTL, with S that stands for gene alternative splicing, and 3' AQTL, with 3' stands for 3' alternative polyadenylation, so 3' APA. So this cartoon shows that we could study the association. For example, on the left, we can see that as the SNP's genotype changes, gene expression changes, while the right one doesn't show the association. So we want to call the left one as an eQTL and the right one not as an eQTL. So hidden variable inference is needed because there are some confounders we cannot directly measure. So hypothetically, the goal is to infer those confounders from the molecular phenotype matrix. So specific in the matrix is an individual by gene matrix. So we want to infer hidden confounders from this matrix. And this task is called hidden variable inference. And in the GTAC consortium, the method used is called PEER, P-E-E-R. And the factors to be included as confounders to remove is called PEER factors. Specifically, in the linear regression setting, we treat one gene's expression as Y and one cis SNP as X. Then we want to consider the PEER factors as covariates in the linear regression, so that when we infer the SNP's effect, we have adjusted the PEER factors. So in the GTAC consortium, here are the tissues we listed, and in the parentheses, we list the sample size for each tissue. So the goal of doing this hidden variable inference, interestingly, is to both remove confounders and also, hopefully, to improve the power of QTL identification. So by power, we measure the number of E genes. That means the number of genes that have at least one significant eQTL. So we can see that here, the number of PEER factors chosen by GTAC is indicated here, like 15, 30, 35, depending on the sample size. And interestingly, empirically, the PEER factor numbers were chosen to increase the number of discoveries, so that you can see that there's some plateau effect in the GTAC study. So my student Heather became interested in this question because she wanted to understand what exactly made PEER factor the state-of-the-art method for GTAC analysis. So in the literature, we also see similar ideas used in the surrogate variable analysis, short as SVA. This method was proposed to adjust for hidden confounders in the differential express gene analysis. So to compare two conditions, we want to find the differential expressed genes. Even though it's a different problem, but it's a similar setting. So in this case, every Y is one gene's expression level, and the predictor X is the condition label. In the GTAC, the predictor is the SNP genotype. So even though they were developed for similar purposes, but surrogate variable analysis is also widely used for the DE analysis, and it has a similar core to find hidden variables from the sample by gene matrix. And besides these two, there's another popular method called hidden covariates with prior. So even though these methods were used in different settings, but so far no benchmarking was done to compare them. So, and a common theoretical core shared by these methods is the use of PCA. We have heard PCA in this session. So PCA, it underlies every method listed here. Then the question is, which method is best? And also, a very big reason why peer became popular method is that in this paper, it claims that its performance doesn't deteriorate as the number of inferred covariates increases. This sounds very attractive because we don't know exactly how many hidden covariates should be adjusted, right? So if peer is robust to that number k, it's very attractive. But my student Heather, she's a very careful person. She wants to verify this advantage because she wants to understand why this could happen. So let's come to our results. So we did simulation studies under two settings, which I will introduce on the next slide, to compare PCA with SVA, peer, and HCP. So this is just our subjective categorization of these methods in 5 aspects: computational speed, performance, which is defined based on the power in our simulation, interpretability, the ease of choosing the number of hidden covariates k, and software usability. So I have to say this is our subjective categorization, but we actually found PCA to be the top performer, which is very surprising. And also on the right, we try to categorize or summarize the input needed by each method. So we didn't say the molecular phenotype matrix because it's required by every method. So basically, SVA requires the specification of the variable of interest. Like in the DE analysis, it is the condition. In the EQTL analysis, it is the SNP. And peer factor requires the input of k, so as HCP. So basically, you have to specify a k to run peer. If you change the k, you have to run peer again. This is very different from PCA. You just do PCA, and you get the other PCs, and then you just specify the k. But the k wouldn't change the remaining PCs. So you just have to run PCA for once. And another thing to note is that both peer and HCP have priors or tuning parameters to specify, but PCA doesn't have it. Another thing I want to note here is that we noticed when peer was used in those EQT analysis, it was actually used in a quite inconsistent way. Some people use peer factors directly as the hidden confounders in the linear regression, but other people use the peer residuals as the hidden covariates. So that's why in our analysis comparison, we consider both usages. So here are the 2 simulation designs we used. To be fair for the method, we didn't have our own design, but we adopted designs from high-impact papers. So the first design is actually from the peer paper, the original peer paper's design. So interestingly, we have to try our best to write the code because the original code wasn't available, but we followed the paper as closely as possible. So this design, nowadays, because the paper was from 2010, it doesn't seem that realistic because the simulated genotype data doesn't have LD, and it has very high minor allele frequency. And also, the original design assumes that most of the effect on gene expression was from trans effect, not cis effect. And furthermore, the number of SNPs is very small, only 100 SNPs in total. So to use more cutting-edge simulation designs, we adopted a design from the SUSY paper, which is about a new fine mapping method from Matthew Stevens' group. And this design is more realistic because the genotype is from GTAC, and the SNP number is, we consider 1,000 local SNPs per gene. So this is a more realistic setting, and also we vary the number of known covariates, the covariates we already measure, like sex and age, those kind of things we have measured, and we vary the number of hidden confounders. So we expect this one to give us a more comprehensive evaluation of these methods. Surprisingly, under both designs, we found PCA to be the top performer in terms of power. So here we measure the discovery power as the AUPRC. And we consider all these combinations of methods, but the first one is the truth, ideal setting, when we use the true hidden confounders from our simulation. And the second is the other extreme, unadjusted. We don't do hidden covariate adjustment at all. So we can see that they set up the upper bound and lower bound on the AUPRC. And for PCA, we consider two versions, using PCA directly on the matrix, individual by gene expression matrix, or we use PCA by first regressing out known covariates from the matrix and then do PCA. So I will skip the details because of time limitation, but the take-home message is that PCA has the second best power just next to the ideal case, which is very surprising. And we see that PCA has the best computational time. So another thing we want to note is that the Peer factors, if they're not applied to the correctly scaled matrix, that they can be highly correlated. Like in this case, even though we hope we found 15 Peer factors, but since they're so highly correlated, we essentially only have one. And also, whether you do center and scaling, inverse normal transformation for every feature across samples, or inverse normal transformation for every sample across features, the results will be different. And we have seen all these different usages in practice. This is another factor we have discussed. And finally, applying PCA and Peer factor to the real GTech data, we found that the PCA factors and Peer factors are either highly correlated, which means that they're almost identical, or the Peer factors are highly correlated themselves, but the top ones are very identical to PCA. So, and in the number of discoveries is also very similar. So this is our conceptual summary of these methods. Like Peer factor is a Bayesian version of factor model, and factor model can be reduced to PCA. PCA is a special case of factor model. SVA is a pure algorithm. It iteratively performs PCA, and HCP has its objective function, but it's very similar to PCA. So finally, we want to say that since people have understood PCA very well, we have many good ways for choosing the number of PCs, but we don't have this tool for other methods. This is another conceptual advantage of PCA. And we also want to say that even though we use PCA to choose a much smaller number of PCs than the Peer factors, but the E-gene number remains quite similar, so we don't lose much. So finally, in the discussion part, I want to say that in our analysis, we found PCA to be faster, better performing, and easier to interpret. And also, PCA has a conceptual advantage because it's both a dimension reduction and the factor discovery method. And with our paper, using a lot of detailed analysis, we think we solved the conundrum that inferred covariates such as Peer factors are often difficult to interpret using known and technical and biological variables. So we have the R package for implementing what we proposed. So this is our paper. I just want to share a reviewer's comment here. It's a transparent review saying that a method can become popular by accident rather than sound statistical argument. So that's why I think when we choose method, maybe popularity shouldn't be the only criterion. And finally, I will say that I will thank my student Heather for leading all the efforts in this analysis. And also, just as a quick advertisement, we had a method for doing p-value free FDR control, and Heather implemented this idea we call Clipper together with the QTL analysis, and we found that because we can get rid of the p-value requirement and the number of permutations, we can be much faster than the fast QTL, which is the current state-of-the-art method for QTL analysis. So thank you.